Good morning, everybody. My name is Abdul Mannaan Giles. I am a medical laboratory scientist. My mentor is Dr. Diana Cochran Black, Chair of the Medical Laboratory Scientists Department with the College of Health Professions. My research focuses on identifying novel biomarkers that can be used to assess Alzheimer's disease. So before I expound on that project, I just want to say that I'm going to refer to Alzheimer's disease as AD. So if you get confused, that's what that means. And I want to give you a brief introduction on what Alzheimer's is. It's a neurodegenerative ailment caused by the buildup of amyloid plaques, which results in memory loss and losing your ability to function independently. With 40 million people being diagnosed with Alzheimer's right now, that's approximately 4 people per second, and by 2030, that's expected to rise to 76 million people. It's been around since 1901, but we have still not been able to find an effective cure yet. There are two hallmark players in Alzheimer's disease. At age 50, our brain goes through some kind of mechanism in which the two proteins that we utilize to remove waste from our brain, they fold a specific way. Those two proteins are called amyloid plaques and tau tangles. So what happens is the mechanisms that we use to remove those proteins when they are no longer needed, we can't remove them anymore. So they form plaques within the neuron as well as outside of it, and it chokes our neurons, resulting in brain loss and eventually the symptoms that we exhibit as Alzheimer's patients. One of the biggest issues that we face with Alzheimer's disease is, one, we cannot detect it early, meaning when we detect it, clinical symptoms have already appeared. Two, the methods that we use to diagnose it are very, very invasive because we have to use a lumbar puncture to quantify those specific proteins that I just mentioned in the spinal fluid, and it's very expensive because we use brain scans to quantify those. So that leads me to my research question. Are there any other kind of fluids that we can examine that will be indicative or unique to Alzheimer's patients that we could diagnose early, that are less invasive and are less expensive? The previous literature has identified two possible molecules that we could target, including microRNAs and proteins. So my specific research focuses on microRNA. So if I have the ability to identify the specific unique expression of microRNAs in the blood of Alzheimer's patients, it would give us the ability to detect it before these hallmark clinical symptoms of Alzheimer's appear, such as loss of memory, vision impairment, and losing your items and things like that. So I want to give you an idea on what microRNAs are. I'm assuming that everyone knows that the proteins in our bodies are controlled by genes. Whenever a protein needs to be expressed, our body produces that gene, and that gene codes for a specific protein. Well, mRNAs control the expression of those genes. In many diseases outside of Alzheimer's, these microRNAs are also aberrant in expression as well. So that is what kind of prompted me to target them as for my investigation. So I had a 4-step process to go about doing this. I looked at some previous literature to narrow which mRNAs I wanted to target. I collected only 5 samples because, of course, it's very difficult to get samples from Alzheimer's patients, and I also collected their age-matched controls as well. And then I used a basic extraction process to take the blood out of those patients and to remove everything except the mRNAs that I wanted to quantify. And then I used a specific machine that I'll explain later on to quantify those mRNAs and to compare them to normal controls. So this is the extraction process. Once we have the blood from the Alzheimer's patients, we separate it and we add chloroform, which destroys every single last thing in that sample except the mRNA. And then we use a machine that looks like this, which is common in pretty much every hospital nowadays. It's called a quantitative PCR machine, which quantifies nucleic acids. mRNA is a nucleic acid. And then we would do some statistical analysis to compare age-matched controls to the AD samples. So, what I found was that total RNA in Alzheimer's patients of the early stage was increased compared to normal controls. I also found specific sequences which were increased compared to normal controls as well. The names are there, but you don't have to worry about those names are somewhat complicated. So here is a graphical representation. So I want to explain something. In the yellow, we have the quantification of total RNA compared to normal controls. Now, the lower graph means there's more RNA compared to normal controls. The higher graph means that there's less. So here, these are the two specific mRNAs that I targeted, and I found that they had a larger amount compared to the normal controls as well, and that was statistically significant as well. So what we can conclude, or what this implicates, is that the mRNAs that I found were also implicated in previous literature as well. Other studies are doing similar investigations as I am, so the more mRNAs that we find in the blood of Alzheimer's patients that are unique, it adds to our growing data bank of novel biomarkers that can be used to assess AAD. And the more that we find, it increases our chances of that entering into the clinical setting. So in an ideal world, what we would be able to do is to treat this as a screening test the same way we treat it for mammograms, colonoscopies, and diabetes screenings as well. Instead of using the methods of lumbar punctures and brain scans, we would do a simple blood test, quantify the specific mRNA, and based on that quantification, it would give you the indication, assess your risk of Alzheimer's onset. Of course, that would have to be around at the age of 50, because that is when Alzheimer's is normally considered to appear. So to build on this future work, we would need to also quantify the protein products and the gene products that these mRNAs control. I would have to do some more tests on a larger cohort that is ethnically diverse from different educational backgrounds as well as different environments, because individuals who have Alzheimer's disease, it depends on your educational status as well as your environment and your ethnic background that can determine your onset of Alzheimer's disease. I would need to do it at different stages. All of my samples were people who have early stage Alzheimer's disease. There are 7 stages of the disease, so I would have to do it at all those 7 stages to see if my specific mRNAs increase or decrease to give us a better idea on the assessment. And of course, use other methods to quantify mRNA, such as immunoassays. So to summarize my study, I would like for you to take away 4 main things: is that the lack of the ability to diagnose Alzheimer's disease causes a lot of morbidity. We do have some new technologies, such as one that I use, that can detect Alzheimer's in the early stage. Some microRNAs have been discovered as a novel possible biomarker for it, and future studies are needed on a larger cohort in order for this to be entered into the clinical setting. I wanted to thank my mentor, Dr. Anna Cochran-Black, who was unable to be here, Sarah Nickel, who assisted me with the PCR process, and all of those individuals who had funded me, including the McNair Scholar Program, the Dorothy and Bill Cohen Honors Scholarship, and the Undergraduate Research Creative Activity Forum.